Continuing on nucleic acid immune sensing receptors and their agonists, we reviewed earlier that activating TLR9 with CpG DNA rapidly stimulates an innate immune response from the B cells and plasmacytoid dendritic cells that express TLR9, and that stimulates the early production of inflammatory cytokines and interferon alpha. And then that leads secondarily to an adaptive immune response over the following days and weeks, where B cells secrete antibodies and T cells activated by plasmacytoid dendritic cells and other dendritic cell subsets differentiate into antigen-specific CD4 and CD8 positive T cells. Now, actually, it's a bit more complicated than that because depending on the structure of the CpG DNA, you can drive the immune response either in the direction of the B cells or in the direction of the PDCs. We talked previously about the CpG 1018 with this sequence. This is a type of CpG DNA called a CpG-B because it preferentially stimulates B cells over the PDCs, and that's due to the phosphorothioate backbone and the relatively simple structure. Now, this is a great vaccine adjuvant. It's in the approved HEPLISAV-B vaccine for preventing hepatitis B. But the type of PDC that's stimulated here, called a P3 PDC, which is very good at driving CD4 T cells to support the B cell production of antibodies, actually is associated with a worse prognosis in melanoma compared to a P1 PDC that we'll come back to. And phase 3 clinical trials of CpG-B DNA in treating patients with cancer have failed. CpG-B DNA and also another type called CpG-C that has the same phosphorothioate backbone have very little monotherapy activity in human advanced cancer. So this type of CpG DNA doesn't seem to have a great future for cancer immunotherapy. There's another type that does. That's called CpG-A. The A is because it preferentially drives PDC activation with production of interferon alpha. This is a different subset of the PDC called P1 PDC, and in patients with melanoma, this type of PDC has been associated with improved survival, and there are early trials of CpG-A DNA in cancer with encouraging results, as we'll come back to. The P1 PDC, again in collaboration with other dendritic cells, preferentially induce more of a CD8 positive T cell response and less of the CD4. So what's unique about the CpG-A DNA? How does that differ from the CpG-B? Well, looking at the sequence of this, you can see G-rich regions on the ends of these CpG-A DNAs. And those poly-G regions form structures called G tetrads that stack into G quadruplexes. These are higher ordered structures. These CpG DNAs spontaneously self-assemble into nanoparticles, essentially. And they have a native DNA backbone, so they can be cleaved during TLR9 activation, and that turns out to be critical for the differentiation of this P1 PDC subset. In addition to that, it turns out that G quadruplexes are actually present in the genomes of all of the retroviruses and viruses that infect humans. So it would make sense that the immune system might have evolved a way to distinguish that type of structure from other CpG DNA structures. And the drug that we'll be talking about in clinical development that embodies this type of CpG DNA is called vidutolimod, where it's packaged into a virus-like particle. But before going on to that, let's come back again to the nucleic acid immune sensing receptors for RNA, because these are also important in the therapeutic development of RNA drugs. Obviously, mRNA protein replacement therapeutics need to avoid activating these receptors if you don't want to cause inflammatory side effects. Now, any of these receptors can enhance an antibody response. It doesn't take that much to activate B cells, and that's sufficient for most infectious disease vaccines. But only TLR7 among these different receptors is expressed in the plasmacytoid dendritic cell and appears able to activate this P1 PDC subset that can secrete high levels of interferon alpha, and that, of course, is critical for driving the CD8 positive T cell responses that are critical for cancer immunotherapy. And so if we contrast activating TLR7 with U-rich RNA to CpG DNA. The similarities are that, again, you're stimulating B cells to differentiate into antibody-secreting cells, and the PDCs are activated. It hasn't been studied nearly as much as with CpG DNA, but there's good reason to believe that P1 PDCs, again, are induced that secrete interferon alpha and can drive a CD8 positive T cell response. Now, in contrast to TLR9, which in humans is not expressed in myeloid cells, in humans, TLR7 is expressed in myeloid cells. And so that is believed to activate these cells into a phenotype that some have referred to as M1-like. The basic question here is whether this activation is going to overcome the immune suppression that's often seen in patients with cancer and is believed to be mediated by tumor-associated macrophages and myeloid-derived suppressor cells. There's not a lot of data on this so far, but ongoing clinical trials should address this because mRNA vaccines to treat cancer are currently in development, some of them designed with U-rich RNA and others with pseudouridine, which would presumably not activate this pathway. So we'll learn from those human trials what approaches are most promising, and early data in early-stage melanoma are encouraging for these approaches. So we can expect to see a lot more data coming out in the future years that will guide the further development of these approaches.